Hello everyone, welcome to 10-Minute Physiology. In today's video, I'm going to give you a short introduction to the retina. So with that, let's give it a go. So I would like to begin by tracing the pathway of light through the eye. So if you were to have your eye, the first place that the photon of light will hit is going to be the cornea. So the cornea is the clear epithelial tissue that surrounds the eye. Then, after the cornea, the photon will move through the anterior chamber and through the pupil, which is surrounded by the iris. So after it enters the pupil, the photon is going to hit the lens, and then after the lens, it'll travel through the vitreous humor, which is a fluid that fills the vitreous body, and then it will hit the retina in the back of the eye. So the retina is where phototransduction is going to take place. And in this video, we're going to be focusing on the retina. So with that, let's take a closer look. So if you were to look at the retina, you would see it would look something like this. So the retina has many different layers, but the main thing that you should note is that at the very bottom layer of the retina is the location of the photoreceptor cells. So the photoreceptor cells are present at the very bottom layer of the retina. And the reason why for this has to do with the fact that the rod and cone cells, which are the photoreceptor cells, require a lot of energy. And since they require a lot of energy, they require a lot of oxygen. And therefore, in order to supply these oxygen and nutrients, these rod and cone cells are present at the back so that they can receive a lot of blood flow. So we basically have two types of photoreceptor cells, as I said. The first are the rod cells, which are responsible for sensing light and dark, so they're monochromatic. And then the other ones are the cone cells, and these are color sensitive. Now, it's important to realize that overall, the number of rods greatly outnumbers the number of cones. But the cones are going to be concentrated in a special region of the retina called the fovea. So it's at the fovea where we have the greatest amount of color vision. So now what we're going to do is we're going to look and see how the rods and cones are distributed across our eyes. So this graph right here shows you the distribution. And what we see here is that at the fovea, we have the greatest number of cones. But as we start moving away from the fovea, the number of cones decreases. And as we start moving away from the fovea as well, what we see is that the number of rods greatly surpasses the number of cones. So the number of cones is greatest at the fovea, and the rods overall surpass the number of cones. So inside the fovea, we have a high density of cones, and this is where we have most of our color vision. Outside the fovea, we have much more rods than cones. So the next thing I want to do is talk about receptive fields. So what is a receptive field? So as you know, the rods and cones are present at the very bottom layer of the retina. And these rods and cones are going to synapse to ganglion cells, as we see right here. So these purple ones are rods, and the blue ones are cones. And what we see here with these rods is that these rods synapse onto ganglion cells, and they further synapse onto these orange cells right here. So what we see here is a property called convergence. So convergence is when one cell receives input from multiple other cells, as we see right here. So what we see here is that as we move away from the fovea, from cell 1 to cell 2 to cell 3, we see that the number of rods or cones increases. And what we see here is the amount of convergence increases. So cell 3 is receiving more input than cell 2. And cell 2 is receiving more input than cell 1. And so what we see here is that the amount of rods or cones that converge onto a ganglion cell is what determines the size of a visual or receptive field. So the key point here is that as we move away from the fovea, the amount of convergence increases, which means that we have an increase in size of receptive fields. This is why cell 3 has a higher receptive field than cell 1. Now, it's important to realize that the size of a receptor field has sort of a drawback to it. In general, as we increase the size of a receptive or visual field, this means that these fields tend to increase in their light sensitivity. In other words, they're more sensitive to light and dark, but they have a lower resolution. But as we move towards the fovea, which causes the receptive fields or visual fields to decrease, we know that the number of cones increases, and what we see is that as we get a lower sensitivity but a higher resolution. So as we move from the fovea to the periphery of the eye, sensitivity increases but resolution decreases. And as we move from the periphery of the eye to the fovea, resolution increases and sensitivity decreases. So now let's talk about rods and cones in general. So rods and cones in general can be divided into two segments, the inner segment and the outer segment. So when light enters a rod or cone, light travels through the inner segment first and then it hits the outer segment. So the outer segment is where photoreception takes place. And what we see is that inside the rod, in the outer segment, we have discs. And these discs are free-floating. And these discs turn over rather rapidly, which means they die and are replaced rapidly. And in the cone cells, we also have discs, but they are instead invaginations of the plasma membrane. Now, both discs in the rods and cones are very important because they not only are the location where photoreception takes place, but because they are in these disc formations, they have a very large surface area, which allows a lot of light to hit them. So the last thing that we're going to talk about is the types of cones. So in general, there are three types of cones. And the first cone is the L cones, which respond primarily to red light. Then you have the M cones, which respond to green light, and then the blue cones, which respond to blue light. So the S cones respond to blue light, M cones respond to green light, and L cones respond to red light. And it's a combination of these three cones that produces our color vision that we have. And we're going to talk a lot about this in more detail in future videos. So what's next for our vision studies? So the next few videos are going to focus on the following things. So we're going to talk about how rods and cones respond to light and dark. We're going to talk about visual transduction, how our eyes respond and adapt to light levels, color vision, and then we'll finish off with something called IPRGCs. So I hope this video helped you understand what the retina is and give you a basic understanding of what it does. And I hope to see you in the next video. Thanks for watching, and I hope to see you next time.